Welcome, dear students. Now we will start our lecture about DNA, RNA structure, and functions. So these compounds are very important, and from our lecture today, you will know more about discovery of DNA, and you will know more about structure and functions of these very important compounds. So first of all, let's start from some very basic questions. And we will start from information. Information that guide development and functional activity of the cell is located in the nucleus. And here you can see a beautiful image of the cell with nice big organelle inside, and this is a nucleus. Information is written inside of the DNA with the help of so-called genetic code that we will discuss later. At the same time, realization of this information occurs with the help of RNA. So we have DNA and RNA. DNA contain information inside and keep it safe, while RNA needs that this is needed for this information to be used. What is the meaning of realization of information? When we speak about realization of information, usually we mean synthesis of the proteins or development of properties of the body. So we can use both of these concepts. For example, when we will study cell and molecular biology, we will mostly speak about realization of genetic information in terms of synthesis of the proteins. But when we will study genetics, we will speak about realization of genetic information in terms of development of certain property of the body. I would like to show you brief but essential concept of information flow in the cell. So let's imagine that this is a cell and this is a nucleus. When you will study nuclear structure in more details, you will see, as you actually see also now, that nuclear membrane or nuclear envelope has certain holes or nuclear pores. That's why here this line is not a constant, but with some holes inside. So nucleus contain DNA. And then information is transferred from DNA that is located in the nucleus into the cytoplasm. And for this transfer of information is used RNA. And then RNA is used as information source in order to build a protein. So if we want to construct something, we need a plan how to do this. If we want to construct a protein, we need a plan that shows us what is the structure of protein, what is the structure that should be created. And this information essentially located in the DNA, and then it is brought from nucleus into cytoplasm with the help of RNA as transport system. So RNA is kind of transport system for information from DNA into the cytoplasm. And this transport system brings us information that is necessary to know how to construct certain protein. Also, DNA is used for transfer of information from one generation to another generation. And this is relevant in terms of parents and children, and also in terms of cell division. Let's imagine that here we have so-called mother cell with genetical information that is located inside of the nucleus, inside of the DNA. And then this mother cell will give rise to two new daughter cells. And each new daughter cell, in order to be able to perform same functions that were possible for mother cell, they need same amount and quality of genetic information. And DNA is responsible for transfer of information from mother cell to daughter cells. So once again, DNA is used for information that is necessary to construct a protein, but also DNA is used for transfer of information from mother cell to daughter cells. Once again, DNA. DNA is abbreviation from deoxyribonucleic acid, and its function storage of information and transfer of information to next generations. When we speak about RNA, that is ribonucleic acid, its function realization of hereditary information. I would say it is involved in realization of hereditary information because concept of realization of hereditary information is very complex. It involves not only DNA, but a lot of other compounds, a lot of other events. So RNA is involved in these events. Let's speak a few words about prominent people who give us knowledge about DNA. Friedrich Miescher in 1969 found an acidic substance in the nuclei of the cells, and the term nucleic acid was proposed. So, at something acidic inside of the nuclei, nucleic acid. Later, Albert Corsell and Phoebus Levene, they established that DNA that is located in nucleus composed of repeating building blocks. And these repeating building blocks composed of several subunits: nitrogenous base, sugar, and phosphate. Altogether, these three components will make one building block of the DNA that we call nucleotide. Later, Francis Crick and James Watson, very famous scientists, they discovered three-dimensional organization of DNA molecules that I think all of you know, this famous double helix. So double helix in which two polynucleotide chains parallelly coiled around a common axis. In simplified way, we can say that it looks somehow like this. And now let's speak in more details about building blocks of DNA and RNA. As I told you before, one building block is called nucleotide, and it is composed of three elements: nitrogenous base, sugar, and phosphate group. Let's consider nucleotides that form DNA. These nucleotides composed of the sugar that is called 2-deoxyribose. That's why deoxyribonucleic acids, DNA. Nitrogenous bases can be of four different types. For example, one type of nitrogenous base is called adenine, but it can be of another type, and it will be called guanine. Another type of nitrogenous base that can be present inside of nucleotide is thymine, and yet another one is cytosine. What about nucleotides that will compose RNA? They have another sugar that is called ribose. That's why RNA, ribonucleic acids, in contrast to deoxyribonucleic acid, DNA. Nitrogenous bases can be slightly different. It can be absolutely the same, like adenine, guanine, but one special type of nitrogenous base that is present in RNA nucleotides is called uracil. And also, it is possible that it will be cytosine. So you see that there is no thymine nitrogenous base, but instead there is uracil nitrogenous base. And once again, this is schematic illustration of one building block of DNA or RNA. And now comes the question how we can create this beautiful double helix with the help of these building blocks. Let's look how this is it is created. Here you see nucleotide of DNA that composed of sugar, phosphate group, and nitrogenous base that is here represented by adenine. This is one so far separated building block or nucleotide. And this is second one building block that looks very similar with the difference that instead of adenine, there is a thymine nitrogenous base inside. And this is yet another one building block that looks, again, very similar, but instead of thymine, there is a cytosine nitrogenous base. And now, in order to construct a helix, double helix, first, we need to construct one chain of it. And now look at these locations. There are connections between sugar of one nucleotide and phosphate group of another nucleotide. And due to these connections, these building blocks are connected into one chain. So in your imagination, you can extend this chain further, simply adding new building blocks and creating connections between sugar and phosphate group. Okay, so now we see how one chain of DNA is created, but what about second chain? For the second chain, we will need additional nucleotides. For example, this one, and then look what will happen. This nucleotide will come in close proximity to first chain, and then a contact between nitrogenous base of one nucleotide in one chain and nitrogenous base that belongs to nucleotide of another chain will be established. You see? Contact between nucleotides, between nitrogenous bases of nucleotides that belongs to different chains. And same situation will repeat again and again and again. For example, here we see another building block. It should be located like this. And contact between nitrogenous bases that belongs to nucleotides from two different chains will be established. And this will repeat again and again and again. Also, we will need connections between sugar and phosphate. And now we have short piece of double helix. So let's imagine that this is one line of the double helix. This is another chain of the double helix. And this is beautiful three-dimensional visualization where this chain, for example, this one, and this chain, for example, the second one. So now you can understand how elementary building blocks organized together in order to produce double helix of the DNA. And if this is clear, I would like to highlight another very intriguing question. You see that thymine nitrogenous base connected here with adenine nitrogenous base. And the question why? Why thymine nitrogenous base is connected specifically with adenine nitrogenous base, but, for example, not with second thymine or not with guanine? Why cytosine nitrogenous base connected with guanine, but not something different? In order to answer this question, we have to go inside of chemical structure of these nitrogenous bases. I would not ask you to remember this structure in details, but I want to show you this chemical structure because this will be very important illustration of the mechanisms why this, for example, cytosine is able to connect with guanine, but not with something else. This is a chemical formula of adenine, and this is chemical formula of thymine. And due to their special chemical organization and interrelationship between chemical organization of adenine and thymine, it is possible to make such contacts that are highlighted here in red color. So this is why adenine can be connected with thymine with two connections, because they match each other in terms of their three-dimensional chemical conformation. Similar situation will be relevant for guanine and cytosine. Due to their special three-dimensional chemical conformation, they match each other, and it is possible to create even three connections between them. So as general rule that you should remember, adenine is connected to thymine with two connections, while guanine is connected to cytosine with three connections. And this is way how one chain of DNA is connected with second chain of DNA. So once again, illustration with chemical formulas, but do not be scared. I will not ask you to remember this chemical formula, but I hope that you will remember simple concept that adenine corresponds to thymine, and guanine corresponds to cytosine in second chain of the DNA. Okay, what about RNA? You know that in RNA thymine is absent, and in RNA, instead of thymine, you have here uracil. You may ask me, okay, and so what? So why we should spend time studying this relationship between adenine, uracil, guanine, cytosine? And in one of my lectures, I will show you how extremely important this is for cell biology. So this very simple concept of matching adenine with uracil, guanine with cytosine is basis for genetic code. If we can say that genetic code is a special language that is used by the nature in order to encode genetical information, this concept that adenine match with thymine and guanine match with cytosine is kind of essential grammar of language that is represented by genetic code. In order to understand the language, you need to know the grammar. In order to understand mechanisms how nature encodes genetic information, you need to know basis of genetic code, and this is actually basis of genetic code. But in more details, I will explain this later, not now. Now we will look what is the organization of genetic material inside of the nucleus. So, okay, now we know that DNA composed of nucleotides that arranged together in one chain, in second chain, and these two chains connected together. But what is three-dimensional organization of this DNA inside of the nucleus? How it is organized there inside? So surprisingly, DNA molecule is so long that if you will stretch DNA of one cell all the way out, it would be about 2 meters long. Imagine, one cell is so small that you cannot see it without microscope, and nucleus is even much smaller. But inside of this very small nucleus, there is a DNA of total length of 2 meters. It is not possible to be like this without special trick that will provide possibility to condense DNA in order to fit very small volume of the nucleus. And this is actually what happens. In order to fit limited volume of the nucleus, DNA make complexes with the proteins for condensation. And complex of DNA molecule with proteins called chromatin. There are several tricks that are used for condensation of DNA, or we can say several levels of chromatin condensation. One level is called nucleosome level. This is a trick when DNA molecule makes circles around spherical shaped protein core. So these units are called nucleosomes. And this provides this will make DNA molecule 7 times shorter. Another level of condensation is chromatin fibers. It is comprised of nucleosomes that placed very close to each other. And now let's imagine that these chromatin fibers will make such loops. This again will provide dramatic shortening of the total of the length of the I will say this in other way. This provides dramatic increase in level of condensation. Yeah, so level of condensation will be increased further with the help of these loops. This is schematic illustration of these loops, and in order to stabilize these loops, you will need special proteins. So additional proteins used in order to stabilize this three-dimensional structure. Here you can see more nice schematic illustration of this loop of condensed chromatins and scaffold components. So radial loop domains represented by chromatin fibers that organized in the loops with the help of special proteins. And this makes a DNA molecule 1,600 times shorter. Another important point that I want to highlight is that this chromatin fiber can have different levels of condensation. Maybe here it is not so good visible, so I will use a little bit another illustration. So let's imagine that I will draw now loops of chromatin fibers. And at some point, they will be very densely packed. But then in some regions, their packaging will be not so condensed. Still, you see there are loops of chromatin fibers that composed of DNA in association with proteins. But there are some regions of very dense packaging of these loops and some regions of not highly condensed packaging of these loops. Okay, let's use classical black color. This is a region of highly condensed chromatin, while here region of not so highly condensed chromatin. Then again, region of highly condensed chromatin. So what is the difference? Why this difference is present? Regions of not highly condensed chromatin considered to be regions with active genes. Regions with highly condensed chromatin considered to be regions with not active genes. So imagine that you have computer, and in your computer you have some files that you actively using now, And also you have a huge amount of files that you do not need at the moment, but maybe you will need them in future. In this case, you will use archivation of those files that you're not using at the moment. So you will not delete them because maybe they will be very important in future, but currently, since you're not using them, you will use archivation of these files and another files that present in your database you will use now. So this region of high condensation of chromatins is kind of archivated genetical information containing genes that are not active in the cell at the moment. This region where chromatin fibers not so condensed, they represent fragments of DNA molecule that contain genes active at the moment. And this we call euchromatin regions, while this region we call heterochromatin regions. And now let's go further. Yeah, and this is summary of what I just explained. Region of chromatin fibers with active and inactive genes have different levels of condensation. Those regions that are not active, they are highly condensed and called heterochromatin, while regions with functionally active genes, they are less condensed and called euchromatin. So, to summarize levels of chromatin condensation, nucleosomes, chromatin fibers, radial loop domain. All these three-dimensional structures that are used for condensation of DNA relevant for period when cell does not divide. When cells start to divide, condensation of DNA will be increased further with the help of special three-dimensional structure that is called chromosomes. So what is a chromosome? Chromosome, basically it is DNA associated with the proteins. But in more detailed explanation, we can say that chromosome is special three-dimensional structure that is used for maximal condensation of DNA molecule. And this is in use during cell division. Yeah, and here you can see this my words illustrated with the help of the text. So this is special three-dimensional structure that is used in cell division. And due to chromosome, maximal condensation of DNA is achieved. Here you can see schematic illustration, very simplified how it works. So chromatin loop domains condensed further and further and further and transformed into this nice three-dimensional structure that we call chromosome. And here is also illustration of all these levels of DNA condensation, starting from double helix DNA. Then you can see schematic illustration of nucleosome, of chromatin fibers, and then further condensation of chromatin fiber will create chromosomes. Let's look in more details on the chromosomes. Chromosome has so-called centromeric region and telomeric regions. Telomeric regions are terminal locations in the chromosome, while centromeric region it is used to hold two I would say two chromatids together. Right? So this is called one chromatid, and this is second chromatid. All together, they form chromosome. And one chromatid is connected with the second chromatid with the help of centromere. One chromatid composed of one DNA molecule and another chromatid composed of another DNA molecule, and they are identical. So one DNA molecule here is a copy of DNA molecule here. So before beginning of cell division, cell needs to make double amount of genetic material in order to equally transfer genetic material to both daughter cells. And in order to achieve this aim of equal transfer of genetic material to both daughter cells, before cell division, cell needs to double genetic material. And that's why also such chromosomes created. Each chromosome contains one DNA molecule that will go to one dotted cell and second DNA molecule that will go to another dotted cell during very interesting process that is called mitosis, cell division via mitosis. But this we will discuss later. So, tips of the chromosomes, they are called telomeres, and these regions involved in the control of number of cell division. So one of interesting tricks that are played by the nature is that with each division, telomeric region of the chromosomes becomes a little bit shorter. And when shortening of the length of chromosome will reach critical level, such cell will not be able to divide anymore. This is something that is called Hayflick limits, limitation in number of cell division that arise from critical shortening of telomeric regions of chromosomes. Let's look on the chromosomes in a little bit more details. So here you can see that each one chromatid composed of DNA molecule and proteins that used to stabilize this three-dimensional structure. So if you will magnify this in one spot, you will see this double helix with nucleotides that are connected together in lines and chains that are connected together due to connections between nitrogenous bases. Chromosomes can be of different morphology. Here you see simplified schematic illustration of different morphological types of chromosomes. The first one, highlighted in red color, type of morphology is called telocentric. This guy is called acrocentric. So you see that in telocentric chromosome, centromere located on the terminal end of chromatids. Acrocentric is such morphological type of chromosomes when centromere located close to terminal end of chromatid. Submetacentric, this is such morphological type when centromere located very close to the midline of the chromatid. And finally, if centromeric region located exactly on the midline of chromatids, we will call such chromosome morphological type as metacentric. Number of chromosomes is a constant parameter of certain kind of organisms. For example, in your cells, in my cells, in cells of all people, normally should be constant number of chromosomes. Majority of cells of human body have 46 chromosomes. Majority, but not all, because there is an exception that is represented by germ cells. They have only half amount of chromosomes, and in one of our further lecture, I will explain you why nature doing this with germ cells. But majority of other cells that we call somatic cells, so we can categorize all cells of our body for germ cells and all other cells. All other cells we call somatic cells. So all somatic cells, or majority of all cells of the human body, have 46 chromosomes. And these 46 chromosomes comprised of 23 pairs, and in each pair, chromosomes are similar. So we call them homologous pairs of chromosomes. And this is schematic illustration. This is illustration, I would say like this. So this is illustration of 23 pairs of homologous chromosomes. You see that if you will compare chromosomes in pair number 1, they will be quite different from chromosomes in pair number 12. But if you will compare one chromosome inside of the pair 1 with second one in the pair 1, you will see that they are very similar. 2 chromosomes in second pair also very similar. So that's why we call them homologous chromosomes. Altogether, there are 22 so-called autosomes and 22 pairs of so-called autosomes, and they not determine development of the gender. And there is one pair of chromosomes that we call sex chromosome. These chromosomes, they determine development of the gender. If there will be 2 X chromosome, this will be a woman, and man has X and Y chromosomes in somatic cells. So all chromosomes together called karyotype. And karyotype comprised of 22 pairs of autosomes and 1 pair of sex chromosomes. In simplified way, we can represent such levels of chromatin condensation. So once again, starting from DNA, then coming up to nucleosome, then chromatin fibers, then chromosomes, and all chromosomes in cell together called karyotype. So genotype is concept that describe all genes together. Karyotype is a concept that describes all chromosomes together. So here you can see a schematic illustration of the cell with nucleus and chromosomes inside. Once again, numbers that you should remember: 44 autosomes or 22 pairs, and 1 pair of sex chromosomes. They all together comprise karyotype of a human cell.